Good morning. Welcome to the Faculty of Science session, Sun and Science, part of our Super and Science series. I'm Dave Stevens, Vice Dean in the Faculty of Science and Professor in the Department of Mathematics and Statistics. So this is a regular series that we offer that gives a snapshot of research that's going on at McGill at different levels. Today we've got some student speakers, which I'll mention more about in a second. But before we get going on the business for today, the provost of McGill sent out an email last night concerning recognizing issues concerning racism in the university and in the academic sector generally. And so I'd like to pass over to Dylan Wong, who's one of the speakers today, who's going to make some comments on that. Thank you, Dylan.
Good morning, everyone. Before we started, we just wanted to take a moment to acknowledge the strike for Black lives, which is occurring today in academia across the world. So today, scientists, mainly in the US, but really across the world, are halting their usual work to instead learn about the way systemic racism is present in academia and the research community, and to craft ways to really address these inequalities. So most of us just learned about it this morning, but we should all take time today to educate ourselves and really reflect on how we can take action on combating systemic racism within our communities and the university context through our actions and through our thoughts.
Thank you very much, Dylan. It's very important recognition, I think. So to move on to the business for today, then we've got four excellent speakers, four student speakers that have been working on research projects in the university from a variety of programs in the Faculty of Science and the departments in the biomedical science group. So after the four speakers, when the presentations are completed, every participant, or all the participants, will receive a message from the host, Preeti, who will ask you to go and join a breakout room. So the breakout rooms will be there for you to discuss with the speakers their presentations. And the speakers will be rotating through the room so that you'll have an opportunity to interact with each of them. And then after that's completed, the breakout rooms will be brought together, and then there'll be a final sum up. So we'll move on to the four speakers then, and the first speaker is Nadia Blostein, who's working in areas in cognitive science. Nadia.
Yes, hi. So hi, my name is Nadia, and I'm going to talk about some undergraduate work that I did at the Computational Brain Anatomy Lab. I don't have control over the screen. Oh yeah, now I do. Okay, thanks. So essentially, it's a neuroimaging laboratory, and so we work with structural MRI data. Structural MRI is a technique used to take three-dimensional pictures of your brain with a very high spatial resolution. And this is an imaging technique that is very useful for diagnostic purposes, like to find brain tumors. But it's also important for research as well, because thanks to the recent advent of open source databases, you have right now currently databases of structural MRI scans of different types of subjects, and there are usually enough subjects to be able to use pretty sophisticated statistical models. So essentially what you need to do, though, in order to obtain any actual data from structural MRIs, is that you need to apply different image processing algorithms to them that kind of help normalize things, and that can also be used to extract quantitative measures of neuroanatomy. So for instance, something's off. Okay. For instance, say you take the cortex of the brain, which is the structure at the surface of the brain, and you subdivide it into a bunch of predetermined regions. So here they're called ROIs, regions of interest. And you take, for instance, ROI X and region of interest Y. You can use image processing algorithms to compute the surface area of those two regions of interest in all of your subjects. So what's cool about this is that this allows you subsequently to use different types of statistical models on your data, because once again, you've basically used structural MRI data you've used processing algorithms on this data in order to extract quantitative measures of the brain. And this is what allows you to use specific models. So I was using a heritability model that uses a twin and non-twin sibling design. And one thing that I wanted to show you is that essentially we were looking at the genetic correlation between pairs of regions of interest between their surface area. So say you take region of interest X and region of interest Y, and you compute both of their surface areas across, say, 100 subjects using image processing algorithms. You can then use this model to look at the genetic correlation. Sorry, the slides are, okay. And the genetic correlation would be basically amongst the genes that control region of interest X, what proportion of them also controls region of interest Y. This can range from 1 to, sorry, I think something's off with my, I made a mistake, I'm sorry. But anyways, genetic correlation can range from 1, which means that both traits are controlled by the exact same set of factors, or it can be 0, meaning that both traits do not share any genetic factors. And so what's interesting about this is that say you're studying a disease like Alzheimer's disease, and you realize that Alzheimer's disease really affects, you know, the surface area of region of interest X in the brain, and you're studying this a lot, and there's a lot of papers coming out about how region of interest X is really affected in the brain, but then you realize that actually region of interest X is also strongly genetically correlated with the region of interest Y, then this may make you realize that you could potentially study region of interest Y in the context of Alzheimer's disease. So this is really why kind of this model is a very straightforward way to look at this. The idea here, genetic factors, is an abstract idea, right? We don't know what genes they are, but we know that they're controlled by similar, factors, which in itself just tells you something about the relationship between the two traits. And this is also an easy model because it doesn't require very large sample size. You can have as little as 150 subjects, which, you know, in math land apparently is not, you know, a lot. It's better to obviously have huge databases, but so and so these are just some results that we got. So essentially for the surface area, once again, of the cortex, which is the surface structure of the brain, and it controls higher order cognitive functions like learning and memory. We found that within every region here that is the same color, so every region of the cortex that's the same color is essentially controlled by the same set of genetic factors. And what's cool here is that it looks very similar to the canonical brain subdivisions to the four lobes of the brain. So once again, overall, this is just like an example of how you can use MRI data in order to, you know, extract certain interesting quantitative measures of the brains in research. And there's tons of other models you can obviously use, and other things you can use the heritability model to look at as well. Thank you.
Thank you very much, Nadia. Nice to see large sample sizes, like 150. So the next speaker then is Laura Meng, who's a student working in neuroscience.
Hello, can everybody hear me?
You're a little faint.
A little faint, okay. Is my video on? I should try to rejoin the call. Okay. Hello, is this better?
Hi.
All right. Okay, so thank you for the introduction. I'm really excited to be able to talk to everybody today about some of the work that I did in my first 2 years at McGill. And my project was using a mouse model to treat anorexia nervosa. So our lab was interested in eating disorders, specifically anorexia. And I think I'm okay. And anorexia is an eating disorder that basically causes the patient to eat less, resulting in emaciation and also lack of weight gain. And a big issue with it is that it has the highest morbidity of all eating disorders and of all psychiatric disorders. It's diagnosed by analyzing these three criteria that you can see here. And some important things to note about it are that in addition to its high morbidity rate, it also has a very high rate of relapse, and currently there's no cure for it. So it's important that we look at potential targets to see if we can develop a treatment. So one potential target that our lab has discovered are these guys right here, tonically active neurons, which you can see in this orangey red color. And they're a very special type of neuron because while most neurons tend to release one type of neurotransmitter, these guys actually release two, glutamate and acetylcholine. So what that means is that they can affect this whole entire circuitry that you see here that has a lot of other neurons and we think is involved in the development of anorexia. It can affect the circuitry very strongly. So what our lab did previously was that we specifically deleted the gene responsible for this neuron being able to produce acetylcholine. So they can't produce any more acetylcholine from mice. And what we found was that these mice tend to be anorexia-like, or they tend to exhibit an anorexia-like phenotype. So how we test that was that we subjected the mice to an activity-based anorexia model, which you can see the schematic here. And what we found was that they tend to, on this model, eat less, exercise more, weigh less, and also die more than their wild type or unmutated counterparts. So with that being said, my role in this project was to basically use two pharmacologics, L-DOPA, which increases dopamine, which is something that we know falls short in the system in our mutated mice, and also donepezil, which is a compound that prevents acetylcholine from being degraded. So that hopes to aim to basically increase the amount of acetylcholine in the system. And I'm going to be injecting these two compounds in mice to see if we can rescue this phenotype. So what we found was that in terms of food intake, firstly, the blue line is our mutants without any sort of treatment. The red line is our mutants with L-DOPA treatment, and the yellow line is our mutants with donepezil. And the black line in all three scenarios is basically wild-type counterpart. They don't have any sort of mutation. They're perfectly normal. So what we see in the graph in the very left is that without any treatment, on every day of the experiment after the first day, without any treatment, the food intake is significantly less. But with either one of the L-DOPA or the donepezil treatments, it actually seems to partially restore food intake, even though it's not exactly the same, especially on the last 2 days of the L-DOPA treatment. But that's pretty promising. We also looked at body weight, which the legend remains the same. But if you look again at the graph at the very left, without any treatment, the difference in body weight is quite large, and it increases throughout the first couple days of the experiment, remains very large. With L-DOPA treatment, we see this decrease in the difference, although there is still some significant days in which our treatment group still remains at a lower body weight than our wild-type group. But with donepezil, what was really interesting is that not only did it kind of decrease the difference, but on the last couple of days of the experiment, it even seemed to be rescuing some of the body weight. So it seemed like donepezil was really appearing to be a very promising treatment. Finally, we looked at the mouse survival curves, or the survival rate. So how we define survival is that we define kind of an endpoint. And that was either when the mice reached 75 or 80% of its baseline weight. What we saw was that without any treatment, at the very end of the experiment, the difference in the number of mice that survived was significantly different and lower without any treatment. However, with L-DOPA or donepezil treatment, at the very end of the experiment, the difference actually wasn't significant. What was even more promising was that donepezil really seemed to have a large contribution in this, and it seemed to be able to help increase the survivability and the survival rate of our mice. So I hope to have shown through all this data that we're able to use these two different pharmacologics to target a mutant model of anorexia, and that will hopefully contribute to our understanding of the neurological substrate of anorexia and hopefully one day contribute to the development of a future cure. So with that being said, I'd like to acknowledge Matthew, my supervisor, Salah, my professor, and of course, all of you for listening. And I'd be really happy to take any questions later on. Thank you.
Thank you very much, Laura. So our next speaker is Morgan Sweeney, who's a student in the Department of Psychology.
Hello. Okay, so my name is Morgan. I'm a recent graduate of the Cognitive Science program at McGill, and this is my project, Magic of the Mind. And it's a bit different from the research you've seen so far, because instead of like scientific research, it's an approach to science communication. So quick overview. I'm going to be talking to you guys about like why this was important, what it is I did, and then what kind of this project looks like now. So first off, what is science communication and why is it important? If we think about the biggest threats kind of facing our world in the future, the climate crisis, AI revolution, even the current COVID-19 pandemic, most of the greatest challenges of our time can only be solved by people who understand science and technology. However, levels of scientific literacy, which is kind of like how well people understand science technology, vary widely both across the world and even across Canada. So it's the job of science communicators and science educators and scientists as well to bridge that gap between science and society so that both citizens and policymakers make decisions based on research that are able to prioritize the well-being of both the planet and the people. Okay, but the typical method that this has been done a lot in the past usually includes like expensive textbooks and really dense academic journals that just aren't accessible to people, both because the language is really complex, it's very jargony, and it's financially expensive. Like most people can't afford $200 for a brand new textbook. However, this isn't the only way to communicate science to people. Research has shown the potential of narrative storytelling as a really powerful tool for learning. They've done studies that show people actually retain information better and are more engaged with the material, which is really important for a subject like science, which isn't easy for everyone to pick up. And one medium of narrative storytelling that's been pretty popular recently is podcasting. It's really engaging and accessible to anyone with an internet connection, as well as it's something you can do while doing something else. So it's just a really effective way to reach people and increasing accessibility. Okay, so what I did was I made a narrative podcast. So for me, I guess the most engaging medium I had ever had growing up was like fantasy. Reading fantasy stories was what I found to be just the most exciting because of the sense of wonder, the plot, and character-driven narrative. So what I decided to do was try to use fantasy stories to communicate scientific concepts to a general audience. And as like a certain type of fantasy, I set my story in the world of Dungeons and Dragons, kind of like the tabletop role-playing game that was like super lame for a very long time, but is cool now because of Stranger Things, to make it even more engaging for myself and the people who I worked with it on, and for the audience to hear as well. So the way I did this was each episode of the podcast touches on a different topic in cognitive science. So like language, memory are some of the ones that you'll hear a bit about later. And I wrote them in as obstacles central to the plot. So usually the conflict or a character would come in like having some language problem or an attention problem. And then what I did to help communicate the science alongside the storytelling was researchers who actually study these topics would come into the studio, and I would interview them, and they'd talk a bit more about like the theories that I wrote in, what some of the evidence we know about them that supports it, so that people who are listening would hear the fantasy story and then the interview, and they'd kind of learn about it through the storytelling and then hear about like the real world research that they could take home with them and learn more about their mind in the long term. Okay, so just to give you an example of what this looks like, I'll talk you through episode 3, where my main character, whose name is Eve, is attacked by a monster who alters her memory to make her more vulnerable and like easier to attack. So the conflict of this episode is Eve has to get her memory back. And to do this, she has to try to recreate as many factors as possible. So she journeys back to the place where she made the memory, tries to put herself back in the same mindset that she was when she made the memory. And this is a demonstration of the encoding specificity principle, which is a theory in psychology that says the more similar your state of mind when you're trying to remember something to when you first made that memory, the easier it'll be for you to remember. So like another example of this is if you like to chew mint gum while studying biology, and like while learning those biological concepts, and then when you sit down and take the exam, you chew mint gum, it's actually gonna be easier for your brain to recall those same biology facts because that mint gum is part of the context that you learned them in. So your context is more similar when you're trying to recall versus when you made the memory. And then in the co-release interview, Dr. Sweeney Sheldon, who is a memory researcher at McGill, came into the studio and explained each key theory of memory that I had written into the episode so that people at the end learned about their own memories and ways that they could improve or just like better appreciate all the hard work their memory does all the time. So what the podcast looks like now, I has released 9 episodes, so 4 two-part episodes and then 1 trailer, which has been downloaded over 1,300 times in 34 countries around the world. So this little map here is actually, like this is the map of like where people have downloaded them that I get from like my podcast producer. So like the more dark and like colorful, the more downloads. The 2 main goals of this project is to teach people more about their minds. I think increasing awareness of mental health and the way your minds work is really important just in general, but also to shift the idea of what science education and science communication can look like. I really genuinely believe that if we want to have a world that is the best world it can be, people need to better understand and want to interact with science. And I think using creative ways to communicate it is a really great way to get people involved. If you're interested in listening to the show or just want to learn more about it, you can go to magicofthemind.ca, or you can just type in Magic of the Mind wherever you get your podcasts. Thank you guys so much for listening. And yeah, if you have any questions, I'll be here after. And these are my citations.
Thank you very much, Morgan. Very interesting. So the final speaker today then is Dylan Wong, who's a student in the Department of Microbiology and Immunology. Dylan?
Good morning again. Sorry. Here we go. So I'll be presenting a project I worked on last summer in Dr. Judith Mandel's lab through an NSERC summer studentship, and I'm now completing my honors project in the lab on a different topic, actually. So checkpoint blockade is a revolutionary new form of immunotherapy used to treat cancer. It harnesses our own immune system to fight off the cancer. And one of the key players in our immune system are T cells. They're constantly looking for foreign invaders. When our cells encounter a tumor, they don't recognize the tumor as being dangerous because it still looks like our own self cells. It doesn't look like a bacteria or a virus. Fortunately, though, sometimes our T cells are able to recognize that our healthy cells have turned cancerous, and then they can try and destroy the tumor. But cancer has been able to evolve to evade this. Tumors can mutate to express immune inhibitory molecules, or checkpoint molecules, which stop these T cells. One of these molecules is programmed death ligand 1, or PDL1, which binds the T cells, killing it and preventing an immune response against the tumor. Now, even though the T cell recognizes the tumor, it can't do anything. So checkpoint blockade simply blocks molecules like this, removing the brakes on the immune system and allowing it to target and kill cancerous cells. Unfortunately, checkpoint blockade is not always effective. When it works, it's miraculous, but it doesn't always work. So here in this graph, you can see growth curves of the tumor being treated with 2 types of checkpoint blockade, the anti-PD-1 and anti-PDL1. And along the x-axis, you see time, and then the y-axis here is size. So as you can see, when the therapy is effective, the tumor is completely eradicated. It completely disappears. But when it doesn't work, the tumor keeps on growing just like as if it had never been treated. So how do we know when checkpoint blockade therapy will work? It's an expensive treatment, so we don't just want to give it to anyone and hope it works. So through biomarkers, molecules which tell us something more about the system than just what was being measured. So the first biomarker for determining if checkpoint blockade will work is the expression of the specific inhibitory molecule itself. Obviously, if a tumor doesn't express it, then we can't block it. The second biomarker is the mutational burden of the tumor. This measures how much of the tumor's DNA has been altered or mutated. More mutated DNA means more mutated proteins or antigens for the immune system to recognize as being cancerous. The higher the mutational burden, the more likely the checkpoint blockade will be successful. So because of this importance of neoantigens or mutational burden in determining the efficacy of checkpoint blockade, we decided to look at the protein terminal deoxynucleotidyl transferase, or TDT, very long name, I know, because of its capacity to add random mutations into DNA. By mutating DNA, it essentially increases the mutational burden of the cell generating these neoantigens. And normally, because of its capacity to add these mutations, its expression is very tightly regulated into the development of only a few specific subset of cells. But from our work in the lab, actually, we found that this protein is erroneously expressed in multiple types of solid tumors, places where it should never be seen. So you can see that there are multiple types of tumors, including CHRCC and PRCC, which are both kidney cancers, that express this protein, which should really never express it. So in order to investigate the role of TDT as a biomarker, I took two different approaches. The first was through the creation of a clinical mouse model. We created tumors that express or didn't express TDT, and then by growing the tumors in a mouse model, we were not only able to observe differences in tumor growth, but also measure how the immune system responded to the two different tumors. So here in the bottom, you can see the growth curve of two different tumors, one expressing TDT, one doesn't, simply showing that TDT expression does not alter tumor growth. The second approach I took in investigating the role of TDT as a biomarker was through bioinformatics. I was looking for genetic differences in human tumors. Here, I looked at individual human tumor gene expression. So in this case, if you look at the graph, each sample is represented by a column and each gene is represented by a row. So in this case, there are around 600 columns and probably around 20,000 rows or so for all the different genes in the human body. So by differentially comparing the tumors that highly express TDT versus those that didn't express it here, we found that there are clear genetic differences between the two subgroups of tumors. You can see in this heat map by the way the two groups look comparably opposite. So while this is only part of the research I conducted last summer, together these approaches have allowed and will continue to let me research the original question: can TDT be used as a biomarker for checkpoint blockade? I just want to acknowledge the rest of my lab, our collaborators, and then the funding for helping me do this research. Thank you.